Good day everyone and welcome to another lesson in Immunology and Serology. I am your lecturer for today, Dr. Supachay Basit, and with me for today's lesson is Josiah Carlo. Okay, so for today's lesson, we will be discussing about adaptive immunity. So adaptive immunity is also known as the specific immunity, and because of the fact that our immune response here is actually specific toward a certain pathogen. Okay, so moving on, for this particular chapter, we will be discussing for a while. Okay, for this particular chapter, we will be discussing differentiation of T cells, stages on the differentiation of B cells. So what are the roles of T cells in adaptive immune response? What are the role of B cells in adaptive immune response? And some of the laboratory procedures for identification of the different subsets of lymphocytes. Okay, and let us have a background discussion of what adaptive immunity is. As what I've told you a while ago, adaptive immunity is also known as specific immunity, meaning to say the response of adaptive immunity is very much specific to each pathogen or microbial agent. Now, one of the most important characteristics of adaptive immunity is that it has the capability to remember prior exposure. So we call it anamnestic response. So for the anamnestic response, the main cells that are responsible for that are the memory cells. Now take note that memory cells exist for both T cells and B cells. Okay. So, adaptive immunity increases response to a pathogen upon repeated exposure. So, that is the reason why, in order for you to achieve maximum response for a certain pathogen, you need to have several dosages of vaccine. So, Josiah, di ba nagpa-vaccine ka, no? Sakit? Okay. So, anyway, it takes longer to become activated, but it is much long-lasting. So, in the succeeding chapter, we will be discussing vaccination or immunization. Okay, so again, adaptive immunity involves T lymphocytes and B lymphocytes. So, T lymphocytes are responsible for the cell-mediated immunity, while B lymphocytes, okay, these cells are responsible for humoral immunity. Okay, so you might be wondering why humoral, because humoral actually it comes from the root word humerus, which is one of the bone marrows in our body. But do not get confused that only humerus is the bone marrow. So anyway, moving on, let's discuss about the T cell differentiation. Okay, so one of the most important steps in T cell differentiation is the so-called double negative stage. Okay, so T cell differentiation happens in the thymus. Okay, so T4 thymus. Okay, so in order to initiate the start of the double negative stage, as you can see here at the cortex, okay, the chemokine is the one that derives the differentiation process. Now, what do you call the cell of the thymus? We call them the thymocytes. Okay, now you listen, baby. Okay, so thymocytes will undergo rearrangement of genes according to the TCRs, or the so-called T cell receptors. So if you will be looking at the illustration here, so your thymus is divided into two different stages. Okay, no, two different parts. So the outer part is the cortex, so the inner part's the medulla. So here the cells undergo rearrangement of genes, okay, so that the TCR will eventually be produced. Okay, so what are the two common types of TCRs? In cases of T cells, the two most common types of TCR are the CD4 and the CD8. Okay, so please, when you actually watching this screencast, I'm encouraging you to open your book as well, because in your book there's a caption of what actually is going on. So anyway, because excuse me. Okay, So because of this rearrangement of gene, the output is the formation of CD4 and CD8. Okay, so if you notice a while ago, in the illustration here, okay, initially there is what you call the CD3 TCR. So what is the CD3 TCR? It's an alpha and beta chains that can or that has the capacity to recognize an antigen. So if you will be looking at the illustration, there are 6 other chains in 3 pairs that assist with signaling when antigen binds to T cells. So in order for us to understand further, let's go back to the illustration here. So again, if this is your thymus, this is your cortex, and then this is your medulla. Okay. Now, the junction between the cortex and the medulla, we call it the cortico-medullary junction. So as you can see, TP, so when you say TP, this refers to the thymocytes precursor. So in the context of hematology, when you say precursor, this refers to young cells. Okay, so this thymocyte precursor will eventually becomes double negative. Okay, now double negative. Now some of the fragment of this particular double negative has actually developed into the so-called gamma delta chain, and some of them develops into double positive. Okay. So the thing is, the positive and negative selection takes place here, okay, through the CD3 TCR. Okay. So this, if positively selected, the cells here that you can see here, you call it the dendritic cell. Okay, so this is a dendritic cell, and from our previous lesson, when we say dendritic cell refers to what? One of the antigen presenting cells. Okay, so if they have, for example, been selected positively or selected to become positive upon interaction with either dendritic cell or macrophages, either of the two are examples of antigen presenting cells. So some of them become CD4 positive, CD8 positive. So these are just singly positive lymphocytes. Okay. However, if you notice, some of them are being weeded out. So the reason why they are being weeded out, so that these particular cells that has not become positive will eventually need to be removed so that they will not interact with self-antigen. We do not want them to interact with self-antigen, because once they have interacted with self-antigen, the possibility of you getting an autoimmune disease is very high. Okay? Now, if the cells become single positive, they either become CD4 or CD8. So these are already mature, and CD4 and CD8 play a very important role. They play very important role in our immune response. CD4 eventually becomes T helper cells. T helper cells, as the term implies, help for both cell-mediated immunity and humoral immunity. Do you know that if you have HIV infection, the virus will have the target site or receptor site for CD4. So do you know that the level of CD4 in your body is supposed to be around 800 to 1,000 cells per cubic millimeter, or in SI unit, it is between 800 to 1,000 times 10 to the 9 per liter. However, in cases of clinical AIDS, the CD4 level is below 200, and that becomes, and if your CD4 level is below 200, that will make you immunocompromised. Now CD8 on the other hand is important because it is also cytotoxic. Is important for cell-mediated immunity. It can lyse target cells. What are again the target cells? Let's review. Target cells are either virally infected cells or cancer cells. Going back again here in this slide, you will notice that if you will look closely, this is the so-called CD3 TCR complex. So based from our previous slide, we all know that positive selection, negative selection, the processes of that selections have actually taken place in this CD3 TCR complex. Now, if you will be looking, there are actually two major chains of the CD3. These are the alpha and the beta chains here. Okay, so the alpha and the beta chains here are responsible in recognizing antigen. Okay, now aside from the alpha and beta chains, there are 6 other chains in 3 pairs. Okay, so they assist with signaling when antigen binds to T cells. So what are these other chains? We have the epsilon, we have the delta, we have the gamma, and then we have another epsilon chain. Okay, take note that the gamma and the delta chains here actually differ when it comes to CD4 and A. So, a while ago, we talked about the double negative state, that some of the thymocytes express to become positive. Okay? So, thymocytes, therefore, being a young cells of our thymus, would have the capability to express either CD4 and CD8, and they also have the capability to rearrange gene coding for the alpha chain. Okay, so positive selection occurs if the TCR is functional. Okay, however, if you will remember our previous lesson, MHC restriction is very important. Okay, what is MHC restriction? Only cells that react with host MHC survive. Okay, so remember, if lymphocytes have not encountered any cells that were presented via host MHC, we call them the naive lymphocytes, and eventually they will not survive. Naive T cells, naive B cells will not survive. Now, negative selection is very important. So as you've seen in the illustration, by the way, you really need to read this book. Okay, so if you've seen in the illustration, you may refer to page 47 of this book. It says here, cells that react with self-antigen are destroyed. Okay, that is for negatively selected cells. Why? If you will not be destroying them, the chance of you getting autoimmune disease would be very high. Okay, so moving forward, once the T cells have become mature, meaning to say they survive positive and the negative selection process, they would either exhibit either CD4 or CD8. So I told you a while ago, CD4 are T helper cells. They assist in antibody production, and CD8 are cytotoxic T cells. They kill target cells, and you know what is the definition of our target cells. Okay, now let's talk about T helper cells. By the way, for this particular module, I will also be including animations in order for you to understand the role of T helper cells and cytotoxic T cells much better. Okay, now remember, T helper cells have CD4. Okay, now CD4, as discussed in our previous lesson or module, has the ability to recognize antigen and class 2 MHC proteins. Okay, so class 2 MHC proteins are found in antigen presenting cells. So what are examples of antigen presenting cells? If you can remember, these are macrophages, dendritic cells, mast cells, B cells, etc., etc. Okay, so the CD4 account for the 2/3 of the peripheral T cells. Okay, now there are 2 types of T helper cells. Okay, so we have the Th1 and the Th2. So the Th1 cells will produce interferon, gamma interferon. We will be discussing this in our lessons on cytokines, but gamma interferon is very important as an antiviral activities. Th1 cells produce also interleukin-2, tumor necrosis factor beta. Okay, so Th1 cells would have the ability to activate cytotoxic lymphocytes and macrophages. So most likely, Th1 cells would be involved in cell-mediated immunity. On the other hand, Th2 cells have the ability to produce interleukins 4, 5, 6, 9, 10, and 13. And one of the most important functions of Th2 cells is that they help B cells to produce antibodies. So they can regulate B cell activity, or in other words, Th2 cells would be responsible for humoral response. Now, you can actually see the difference, right? Th1 cells for cell-mediated immunity, Th2 cells for humoral immunity. Now, you can just imagine that our T helper cells is actually one of the most important cells of the immune systems, and this is being targeted. These cells are being targeted by HIV. Okay, so can you just imagine the repercussions? If T helper cells diminish, we become immunocompromised. Then we get opportunistic infection. And if our T helper cells fall below 200 cells per cubic millimeter of blood during HIV infection, we have officially entered the clinical AIDS stage. Okay, so do not get HIV. Okay, now there are other T cell differentiation. Okay, so we have the Treg, not T-Rex, Treg. Treg stands for the T regulatory cells. T regulatory cells have both CD4 and CD25 as the receptor. The Treg regulates, which means that they can suppress the immune response to self-antigen, and at the same time, they can secrete inhibitory cytokines to inhibit proliferation of other T cell population. Okay, now, so as you can see, aside from the CD4 and CD8, there are other subsets, okay, of T helper cells. So we also have the Th1 and Th2, which we have already discussed, and then we have the Th17, Treg, and Th9. Now, as you can see, for cell-mediated immunity, we have the Th1 and Th17, and then for humoral immunity and also for anti-parasitic response, we have Th9 and Th2. Okay, and you may want to study this particular illustration, and this is found on page 50 of this book. Okay, so again, I will not be giving you a copy of the PowerPoint slide. I will read the book. So this is Clinical Immunology and Serology by Stevens and Miller, 4th edition. So this is our official textbook for this particular screencast. Okay, moving on now. So we're done with the T cell differentiation. Now it's about time to discuss B cell differentiation. B. Why B? Primarily because originally the reason why we call it B, because of the bursa of fabricius. But since we are not birds, we do not have bursa. We have the so-called bursal equivalents. So our bursal equivalents are either fetal liver when you were still a fetus, or bone marrow now that you are an adult already. Okay. So B cell differentiation occurs in bone marrow. So if it's very important that you recall your lessons in Hematology 1, because B cell differentiation starts with a hematopoietic stem cells that develops into early lymphocytes progenitor. Okay, so when you say progenitor, it means that it is the mother of all lymphocyte cells. Okay, so they are the ones that produce the progenitor. An example is lymphoblasts. Okay, so B cell differentiation involves developments of mature immunocompetent B cells. Take note of the word immunocompetent. Okay, meaning they are competent as far as the context of immune response is concerned. Okay, now these particular B cells need to be activated by an antigen. Otherwise, they become naive. Okay, so once activated, okay, B cells can be differentiated into one of the most important cells again of our body, and this is plasma cells. Why important? Because plasma cells eventually will help us. Plasma cells eventually becomes factory for antibody production. So, let's discuss now the B cell differentiation. So, in this particular process, we have the so-called antigen-independent phase. So, this is pre-B cells, immature B cells, and mature B cells. Okay, so, as you can see in the illustration, at the early stage of B cell development, particularly during the so-called antigen-independent phase. By the way, the reason why we call it antigen-independent phase is because these B cells have not encountered antigen yet, okay, while they are in the bone marrow. So, of course, the lymphocyte progenitor is the stem cell, and the stem cell actually interact with the so-called bone marrow cell. So, this is the bone marrow stromal cell. So, the interaction, as you can see here in the c-kit, will trigger the development of pro-B cells to that of the pre-B cells. Now, if you will be reading your book, it says here in your book, in order for the B cells to develop, several growth factors are needed. So in your book, it is written, these are the E2A, the EBF, or the early B cell factor, interferon regulatory factor, or the IFR8, and the pair-bax proteins, okay, or the PAX5. Okay, so in addition, interleukin-7 is also important for this development stage. Now, as you can see, why c-kit is important? It says here, c-kit, if you want, you may proceed to page 52 of your book. It says here, c-kit is a receptor on the pro-B cell. It interacts with a cell surface molecule called stem cell factor on stromal cells. So this is very important because the interaction will eventually trigger the activation process. Okay, so once pro-B cell develops in the pre-B cell, so you would see that one of the indicators that it is already a pre-B cell is the presence of the mu surrogate light chain. So here, okay, so the presence of the surrogate light chain here is actually brought about by the accumulation of the mu chains in the cytoplasm. So once there is enough accumulation of mu chains, so this will be expressed, okay, at the surface of the cell. So that's the reason why we have the so-called the mu and surrogate light chain. So alongside with this, so we also have the alpha and beta chains. So all of these alpha and beta chains, the mu and surrogate light chains, all of these, okay, are collectively known as the pre-BCR. So when you say pre-BCR, that stands for pre-B cell receptor. Okay, so the pre-B cell receptor, eventually, when these are formed together, you now have the so-called stage letter C, the immature B cells. And how would you know if they are already immature B cells? Okay, again, the presence, it says here in your book, that immature B cells, okay, are distinguished by the appearance of complete IgM molecule on the cell surface. So there you go. Okay, however, if self-antigens give negative signals, okay, so for example, the presence of self-antigen, it gives negative signal, these particular B cells will need to be eliminated. So that particular process, again, of the programmed cell death is called apoptosis. This is very important so that you do not want these B cells that have negative signals to remain because they interact with self-antigen, primarily because this will make their body to have autoimmune disease. So it's very important that these cells are eliminated. Do you know that about 90% of the B cells never leave the bone marrow because of this process? Okay, 90%. Can you just imagine the figure? Even if they are capable of producing antibodies, but if they give negative signal, okay, so they will need to be eliminated. So this particular process of eliminating B cells that are capable of producing antibodies, we call it central tolerance. Okay, only about 10% will enter the spleen or the lymph nodes. Okay, so if the B cells enter the lymph node, so you'll see that they have CD23, IgD, IgM, alpha and beta chain. So they will be the so-called follicular B cells, and they are the one that enters the circulation, while those that have CD1, IgM, alpha, and beta, okay, will become marginal zone B cells, and they are the ones that remain in spleen. Okay, so that is in a capsule. That is the differentiation of B cell. Okay, so let's discuss again the cells, each of the cells one by one. So let's start with the pro B cells. Okay, so again, as what I've mentioned before, pro B cells develop from rearrangement of genes that code for heavy and light chains of antibody molecules. And the reason why pro B cells are made, because it is made specific for certain antigens by the variable region. Okay, so it is the pro B cells must successfully rearrange one set of heavy chain genes in order for them to become pre-B cells. So as seen here, okay, one set of heavy chain gene in order for them to become pre-B cells. Now, once they are already pre-B cells, it is expected that they have the heavy mu chains of the IgM class. And the reason why the heavy mu chains of the IgM class appear, okay, on the cell surface, because primarily the mu chain accumulates in the cytoplasm. Once they appear on cell surface, okay, so they already have the so-called surrogate light chain. So again, aside from the heavy mu chains of IgM and the surrogate light chain, the B cells, the pre-B cells, must also contain short chains of IgA and IgB. So collectively, we call them the pre-PCR. Now, in order for them to be recognized as immature B cells, they must have complete IgM antibody molecules on cell surfaces. Okay, so this complete IgM antibody will serve as antigen receptor. But they must be careful not to interact with self-antigen, because once they have interacted with self-antigen, they will be eliminated. So this particular IgM antibody would also have variable regions. By the way, you may, in our next lessons, in the succeeding lessons, I will be discussing the different parts of the antibody. So you will notice that some of the antibodies part are called constant region, variable region. So we will be discussing these parts more in detail on our succeeding lessons. Anyway, so the IgM antibody on the cell surface have variable regions, and it's very important because the variable regions will determine the specificity for antigen. This way, the antibodies that we will be producing will only be specific for a certain antigen. Let's say, for example, you develop antibodies against SARS-CoV-2, the causative agent of COVID-19. That antibodies will only work against SARS-CoV-2 but not against hepatitis B. That's what you call specificity. Okay, so the immature B cells may also contain CD21, CD40, and the class 2 MHC molecules. Okay, so the immature B cell have recognized the process of central tolerance. Again, I've explained this a while ago, because central tolerance involves elimination of almost 90% of B cells that have self-reactive receptors. Okay? If immature B cells survive the process of elimination of central tolerance, now they will now move forward to the spleen. Okay, in the spleen, if they develop as marginal B cells, they remain in spleen, which means that they have the ability to respond quickly to blood-borne pathogens. Okay? Now, those that will not have migrated to lymph node and other secondary lymphoid organs, they will be recirculated in our body. Okay? So, pala yung mga pinagdaanan ng B cells. You can just imagine the process. Okay? So, mature B cells, what are the other characteristics of mature B cells? Mature B cells have the ability to exhibit IgM and IgD. So, by the way, these are classes of immunoglobulins. Okay? The surface immunoglobulins provide activating signal to B cells when comes into contact with antigen. Okay, So when stimulated, they enter the so-called antigen-dependent phase. Okay? From antigen-independent phase, we are now at the antigen-dependent phase. So the antigen-dependent phase involved the formation of memory cells and antibody secreting plasma cells. Okay, so B cell differentiation involves plasma cells. So this is now a mature plasma cells. So you will notice that mature plasma cells would have lack of cytoplasmic immunoglobulin as shown here in stained plasma cells because of the presence of the vacuoles, but little or no surface immunoglobulin. And then, so let's discuss now of plasma cells, of mature plasma cells. They have the ability to produce antibodies, and they are found in peripheral lymphoid organs and bone marrow. T cells in adaptive immune, the process of immune response, APCs or antigen presenting cells are activated during innate immune response. So what are the APCs? Macrophages, dendritic cells, for example. So T cells interact with APCs to initiate adaptive immune response. Okay? T cells circulate throughout the bloodstream, lymph nodes, secondary lymphoid tissues looking for antigens. So they are like police patrolling our body. Okay? So here's what will happen. So as you can see here, this is an example of macrophage. A macrophage is an example of antigen presenting cells. So here, macrophage have engulfed bacteria. So what will happen is that this macrophage will digest bacteria. Okay, so that's an example of non-specific immunity, right? So macrophages will now display through the MHC class 2. Remember, MHC class 1s are found, class 1, okay, this is found in all nucleated cell. MHC class 2 is found in antigen presenting cells such as macrophages. So MHC class 2 displays the bacterial fragment. So again, if it's MHC class 2, it will display the material, the bacterial antigen, and it will display it for what? For TH cells. If it's MHC class 1, it will display it for the CD8 or cytotoxic T cells. Okay, so MHC class 2 display it for the TH cells. So what will happen? Because of the presence of the cytokines interleukin 2 in particular, the T helper cells will be activated. Okay, so some of the T helper cells will be activated as memory cells, and some of them will be activated as the plasma cell CD4. Okay, so what will happen is that the CD4 T helper cell will activate B cell to become plasma cell. Like CD4 is not plasma cell. Sorry, I got confused a while ago. CD4 or T helper cell. Okay, so T helper cell, the cytokines here will allow B cells to mature so that B cell will now produce antibodies that is specific to this particular bacterial antigen. And then cytotoxic T cell is important also because the cytotoxic T cells will destroy target cells. Okay, macrophages and neutrophils will further be involved in inflammation. Okay, now how about the cytotoxic T cells? So cytotoxic T cells will be encountering MHC class 1. So MHC class 1 which means that they are found in all nucleated cells of our body. Imagine you have a virus. Okay, let's say the COVID-19 virus, or we call it the SARS-CoV-2, enters the cells of our lungs, alveolar cells. So the alveolar cell is now the target cell because it is already virally infected. So what will happen is that the alveolar cell, which is now a target cell, will supposed to display viral material or viral antigen because all of the cells of our body except for the antigen-presenting cells, are supposed to have MHC class 1. So, you can see that yellow thingy here, that is already the viral fragment. So, what will happen is that MHC class 1 is displayed to CD8, which is the cytotoxic T cell, and there would be interaction. And what will happen to the interaction? Now, this cytotoxic T cell, okay, will now produce two main things, okay? First, it will produce granzyme. And then, it will produce perforin, okay? Oh no, sorry, perforin first, and then granzyme. What is the purpose of perforin? The perforin will create hole in the target cell. And once hole is created, granzyme again is produced, and then granzyme will pass through, causing the alveolar cell to die. Okay, so that's the action of the cytotoxic T cell. So, perforin granzyme system is the one that is produced by CD8 cytotoxic. Okay, how about the role of B cells? There are also what you call T-dependent antigens. Okay, so again, the illustration that you have here is actually the enlarged version of this one. Okay, so activated T helper cell will allow B cell to mature. So to discuss this in detail, okay, so the T helper cell, so the B cells are supposed to have the CD40. This will allow them to interact. So the T helper cell, so now the T helper cell will now produce, the B cell becomes plasma. They will now produce antibodies specific to the antigen or to the bacterial pathogen, for example. There are also T-independent antigen, and most of them are gram-negative, such as this one. So gram-negative bacteria, meaning if it's T-independent antigen, there is no need for T helper cell. But if it's T-dependent antigen, there is a need for T helper cell. Okay, so what's the importance of knowing the subset of lymphocytes? Usually, guys, HIV patients will have to undergo the identification of the subset of lymphocyte by means of flow cytometry, which is another lesson here in immunology. So it will segregate lymphocytes into different subsets and is used via monoclonal antibodies. So it's very important for AIDS patients, for assessing or evaluation of immunodeficiency disease, and for assessing malignancies. Okay, so this sum up our